All right, great. And are we recording this then?
Yes, Celia, the recording is on.
Deepsha, are you starting the recording? Okay. Yeah.
Yes, it is already recording.
All right, great. So welcome everyone. Welcome back to Genomics in the Virtual Lab. We'll continue to speak today about research literature, genomics and virology, microbial communities, and antiviral drugs, vaccines, and antibiotics. So we'll talk about analysis of genomic sequencing. We'll talk a little bit more about next generation sequencing today, and we'll talk about it in the context of virology, the study of viruses. And quite a number of different examples that are probably going to be interesting for all of you to see in the context of what's going on around the world. And hopefully you will have some good questions to talk about how next generation sequencing is going to be applied or useful in the study of viral genomics. So let's go back to the weekend activities. I know that Sanjali has submitted a video and Abidur has submitted a presentation. So is either one of you here today to speak and present your slides just so that you can practice that and show us what you have? All right, Sanjali, why don't you unmute yourself and you can share your screen. We'll briefly take a look at what you have.
Hi, can you hear me?
Yeah.
Okay, so for my presentation, I did Leber's hereditary optic neuropathy. And so first I found the UniProt accession number, which is the P03915. And then I found that it was a reliable entry because it belonged to the SwissProt section of UniProt, meaning that it was reviewed and also had a yellow reviewed star. And so about this disease, LHON is a maternally inherited disease, which means that there's a mitochondrial pattern of inheritance, and this means that females with the mitochondrial DNA mutations will pass it down to their children because only egg cells contribute mitochondria to the embryo. And this disease causes central vision loss, and symptoms start off as blurry and cloudy vision, and it worsens until vision loss is permanent. So some patients also experience cardiac conduction effects, which are irregular electrical signals that control the heart, and neurological disorders. And this occurs due to mutations in the MTND5 gene, and the chromosomal location is in the mitochondria. So this disease is implicated in 3 different diseases, or this gene is implicated in 3 different diseases. So the first, as I mentioned, was the Leber hereditary optic neuropathy, and the next one is Leigh syndrome, and the third is Meles syndrome. And then I included the OMIM IDs for all 3 of them. And I did find a structure for this protein. There were actually 4 entries on PDB, so I just listed all 4 of them here. And these are the PFAM domains and the session numbers for each. So from PDB, I used the 5xTD structure. I actually wasn't really sure which one to use, so I just stuck with one throughout the whole project. And I found the biological assembly, and these are the ligand interactions. And then I found a chart that explained the family, superfamily, and fold information. And then finally, from Pymol, I got an image of this structure. And I noticed that for my disease, there were actually 3 mutations. So I mapped out one of them here. So which was at like position 171, where the change was from alanine to valine. So this is right here. And then the last thing I did was find the electrostatic image of my protein. And these are my resources. Thank you.
Okay, so just a quick note on your previous slide when you're showing the mutation, do you know how to describe the mutation, how to just mention what it is?
Like, do you mean the, like I
No,
said it was from...
Yeah, when you have the arrow, right? So when you have a mutation, you will say, you know, whatever is in the reference, then the number, the position, and then whatever the changes to. So that's just how you name that mutation.
Yeah, okay, I just said that. I didn't put it on the slide, so I guess I can add that.
Okay, Great, excellent. Looks really nice. And what we're doing with all of these presentations that you submit, once they are in a format of a video, we're actually sharing them with Dr. Sona Vasudevan, and also we will have at the end some external people that will join and have additional comments on your presentations. Those will include presentations of these projects as your activity for week 1, and will also include presentations from your final project. Okay, sorry, did anyone else want to share now that hasn't shared already? Yeah, thank you, Mohit. Okay, so if there's no one else, again, we hope that all of you will have the chance to submit your presentation. If you have a presentation like we saw today, Sanjali's presentation, just record it. We yesterday discussed how to record a presentation. You can do it in PowerPoint, you can do it in Keynote, you can do it in QuickTime if you have a Mac. So really, it would be a way to submit your presentation for somebody external to review, because when they just look at your slides, they don't know, what exactly are you showing, and so there's some explanation that you have to do. And that explanation in a video format will actually be reviewed. Okay, so we will continue speaking today, and let's just review briefly what we talked about last time. We have quite an agenda for today and for the next few days. So we'll talk about how to generate next generation sequencing data, what types of formats for next generation sequencing data we have beyond FASTA files, which you're already familiar with. We'll talk about how those mutations can be categorized that you found, for example, a mutation. How do we actually categorize those mutations? And how do we use next generation sequencing to study outbreaks of viral pandemics or viral diseases? And we'll also talk about kind of introduction in metagenomics in general. What is metagenomics and how does it work in terms of next generation sequencing data analysis? So I'll have a few examples. I saw that quite a number of you have been able to start looking at the coursework in Introduction to Genomics and Genomics 1. We've added a couple more courses for you to start looking at this week, Introduction to Metagenomics and Metagenomics 1. I'll briefly introduce those today. Last time, one second, so Parimala is asking, please, if you have any questions, you can just put them in public. If a protein does not have a mutation but gets overexpressed, and what is the reason for the overexpression? So I'm not able to find any OMIM ID, I think. Should I proceed further or should I not? Okay, so expression of genes or proteins that they produce is part of, it could be a result of a variety of reasons. For example, some genes are controlled by promoters. Most genes, they contain a promoter, and that promoter could either be accessible or inaccessible. It could be blocked by a microRNA. Some genes could be in an area that is tightly packaged in heterochromatin and will not be accessible. So there are a number of factors as to why some genes become expressed, overexpressed, underexpressed, or blocked completely. Those are typically in the study of transcriptomic and epigenetic data. We'll talk a little bit about those two types of data, but if you do have a protein and that protein is overexpressed but does not contain a mutation, you can definitely do your presentation on that. You can explain what is the role of that protein, what function does it have, what pathway might it have, and you can also get a lot of that information from the gene, not necessarily just the protein. Now, if a protein has a mutation, we'll talk today about a few examples where that mutation will cause that protein to not properly function or not function at all, even though it is expressed. So we'll talk about that a little bit more today. Now, I do recommend that just in the context of what happened last week, you stick to whatever was the assignment. And the assignment was related to some of these genetic diseases that do contain a mutation. So if you can find something that is more aligned with that assignment, I think that would be better for a submission. But for your project, which is what we're talking about through the rest of this week, I think you can go ahead and focus on that example. I don't know which example exactly you have. Okay, so let's review again kind of some of the things that we have been covering in our last session. Paramala, is that? Okay, great. Just making sure. Again, if you could switch your chat to not be private but public so that we can all see there, including Bibsha and Mohit. If I missed the chat, you can let me know. Okay, so last time we had introduction. Sorry, Kylie is saying that she wants to do the genomic courses next week. Yeah, you can definitely, you will have time to still work on these next week. I'm just making sure that everybody kind of knows what's in those courses. So what we talked about last week is how mutations at the nucleotide level correspond with amino acid level. And to detect those mutations, there's a number of bioinformatics pipelines. So this week, we'll be talking about genomic data, next generation sequencing data. That data has to be prepared for analysis because before we can actually identify those mutations. So in some cases, we do a variant calling pipeline. That variant calling pipeline is explained in Genomics 1. The objective of a variant calling pipeline is to identify variants, in that case, specifically associated with somatic mutations. Somatic mutations are not the inherited kind, not the ones that you inherit from your parents. Those are called germline mutations, but those are variants that start appearing over time, eventually leading to some disease. For example, in cancer, many genes are designed to produce proteins that can suppress different processes, that can fix the mutations that occur on the DNA, that can make the cell continue to behave normal as part of the same tissue. When it becomes abnormal and starts behaving like a tumor instead of a tissue, it actually could be a result of accumulation of mutations called somatic mutations, somatic variants. To identify those, we have to have a sample of the person that the tumor came from that contains both the germline mutations, which are the differences, genetic differences that that person has from their parents, as well as the somatic ones. So typically you would end up with two separate samples, one from the blood, for example, from the normal, and one from the tumor, which is going to give you the ability to differentiate between the two and identify only somatic mutations. And an example of that is provided also in Genomics 1, where in the P53 gene, we can see a mutation, which is a SNP, right? Single nucleotide polymorphisms. Now, today we'll dig a little bit deeper into how we can actually identify those types of variants, because when we talk about next generation sequencing, that produces short reads which have to be aligned to the reference genome. We talked briefly about that last time. We'll talk more about that today. In the case of viruses, it's the same story. We have here the sequencing with a lot of high coverage of reads for some genome. In this case, this is the COVID-19 genome. And this COVID-19 genome, you can see it has about 30,000 nucleotides. And the reads, the information that we actually first have access to, are the sequences of these short reads. In the end, when they are processed and mapped correctly onto the reference genome, we actually get the FASTA sequences that you are already familiar with that we talked about last week. So those FASTA sequences are the consensus sequences if the data originated from next generation sequencing. We also talked about choosing a topic for your project for this week. So I just wanted to remind you what are some of the considerations around selecting a topic and then working on your topic throughout this week. Hopefully you will have the time, because at the end of this week, or by the end of this week, maybe after Sunday, our reviewers are going to start looking at your project. So as you think about some of these, the topics that you already started working with, or other things that you're interested in, you can select the topic, and I'll walk you through the process again today. So first of all, you have to select the topic that you know and care about. Okay, and Linnaeus, yeah, if you have an outline for a specific disease that you're interested in for last week, right, you have a very specific assignment, and you should have gotten that assignment again as an email. So that is just to submit from last week. If you want to continue working on that project, you can expand that over the course of this week. I understand. Yep. Okay. So first, you should select the topic that you know and you care about. So the question is, can you prepare a few slides explaining the topic background? And can you see yourself spending a few months researching the topic? So it would be great if all of you can start either building on top of what you already did, because obviously you were able to prepare a few slides, or if you don't see yourself interested and passionate about the project, think about something else. You already know how to prepare kind of background slides about some element of your project. Now you can apply it to a different topic. The other part of it is that you want to find something that is interesting to your audience. Again, so we talked about this a little bit last time. This is not necessarily a requirement for this specific project, but something to consider overall. You can actually think about how many people care about the topic. Or does your audience care about the topic? Or do you hear this being discussed and people worried or concerned or interested in the topic? Finally, you have to find something that has sufficient data. You probably noticed that if you choose any topic, that doesn't necessarily mean that you can actually find all of the data that you need, right? So sometimes you have to find a topic and then change the topic if you don't have enough data. Okay, so who has already submitted or who has already thought of a project? Okay, regarding our project this week, are we going to do a review of available data of a disease of our choice? Yes, so again, we'll talk about what to include. You have to have an introduction. In that introduction, let's actually talk about that briefly, right? So, okay, so that should be your project. So how do we actually start working on a project proposal? Okay, so working on a project for your proposal, you start with a general background, which means that you do a review of available sources and you categorize those sources. This not necessarily has to be a part of your introduction, but what you want to do is you want to start from a broad overview. So just like you're mentioning there, Suparna, a review of available data, or maybe publications, top publications. And when you do this review, you start focusing on some unanswered questions or maybe difference in opinion, maybe a debate. I'll give you an example. Recently, related to COVID-19, there were a lot of mentions of a particular mutation. That mutation appeared in the spike glycoprotein, and it was the change from a D to a G in the position 614. So some people have just noticed that this mutation started emerging in a lot of patients. Others suggested that this mutation could lead to an increase in pathogenesis, so how disease develops over time. But others have suggested that these mutations can appear for a variety of reasons. Some of them appear because the samples are prepared in tissue culture, and in the Ebola virus outbreak, that has been noted that some mutations only appear in tissue culture and not directly in patients. Others have suggested that mutations could be non-related to symptoms. They could just appear because it does not play any kind of a role for the virus, just mutations appear over time. And recently, there have been some publications from, I believe, Harvard Medical School that are still not peer-reviewed, however, that suggest that this mutation is associated with increased infectivity. So there you go. You have a difference of opinion, right? You have some subject, which is a mutation in a particular protein. Then you have difference of opinions, what might be the significance of this mutation. And you want to add to whatever those opinions are. You want to maybe confirm or disprove their hypothesis. So you really identify your hypothesis as you are doing this review, right? And at the end, what you have is you have your question or hypothesis that you want to start with. Okay, so you start with a broad overview, you select some specific topics that are being discussed, and there's maybe a difference of opinion or some interesting aspect that catches your attention, and then that's how you define your question or hypothesis. Does that make sense? Or does that answer your questions, Suparna? Okay. So as a result, what your end goal is to create a poster. Think of it as a poster. So if you are, still a student, perhaps, maybe you haven't had a chance to present a poster at a conference. What is a conference? A scientific conference is when like-minded people around a particular topic come to discuss research, right? So they have presenters that speak about their current research, and they have students that are perhaps working on projects. Those projects might not be there yet. They might not be complete in the sense that ready for a publication. Maybe they are still at the stage that you're at. They're thinking about projects, they have done some literature review and some analysis, but the conclusions are not strong enough for them to go after a publication. So how do they present their projects in the meantime? They actually present them in a poster format. A poster is like a stepping stone to an actual publication where you present what you have done and get feedback from the participants of that conference. So in a sense, this program, you can view it as a conference. We have some people that are experts in their domain. They come and share with you their research or from their experience. Then you get your peers that are also working on different types of projects. You present to everyone, you get feedback, and the feedback is supposed to help you continue work on your project in a particular direction. For example, once you present this project, somebody might come and look at it and say, okay, this is a very interesting analysis, but have you actually looked at this? Have you asked this question? Have you thought of this? Did you see this publication? Okay, so that kind of feedback can cause you to look deeper into some element of what you have been working on and start thinking, okay, what do I do with this next? And maybe one of the next steps for you is to actually develop a publication, okay? But many times that requires an element from each one of these 4 different things. You can see here, the green section is introduction. Then you have the red section, which is the methods. Then you have the blue section, which is the results, and the purple one, which is your conclusions, okay? So right now, we are just preparing for doing all of those. And to help you actually do that, we have a form on the project submission. So Bibsha just posted a link. Let's take a look at that link. Okay, so here's that link. Oh, sorry, reloaded. Okay, so project submission. It's a form that is dynamic. So actually you put in your name, you put in your information and your information about the project, and you will see it updated if you want to change something. So just as an example, here I'm submitting a project for me. You have to identify a project title and a problem statement. So the project title is a short sentence that describes the title of your publication, your poster, your project. That is going to be your title. Then you have the problem statement, right? The problem statement is a little bit expanded definition of what you are trying to solve. Project summary will include both a few sentences on the background, a few sentences on what you are planning to do, and what you are expecting to achieve in this project. We talked about the different types of data. So typically you're not going to be generating your own data, you're going to access somebody else's data. So that primary data, that raw data that you want to reference will be pasted right there. And your literature review will come in here, reference publications. Okay, so all of you should be able to see this post on the forum. Do you have any questions about this form? Does anyone have any questions about this form? Okay, does anyone see this form? Do you see this form on the forum? Hello? Okay. Yes, yeah, so that's exactly what I was saying. This form is dynamic. So right now, I can change something here. For example, I'll add a project title. So SARS-2 in autoimmune conditions. Okay, let's say this is my submission. I go down here, I click on submit. Okay, after it has been submitted, I can reload. Okay, and now you will see that my project title is already here. Okay. All right, has everyone else seen this form? Okay, great. Karina. All right. All right, great, great. Thank you. That really helps me know that you are following along. Okay, another thing on the forum, you'll see here that we start pasting additional information. So project submission is going to be your primary topic right here. Okay, so you can go here, we'll always be on top. Now you can also see that we've added a few of the top performers. For those of you that want to have a profile that stands out, okay, you can see that we've started highlighting some of those profiles. So we think that a lot of you are doing great. How do we know that you're doing great? We're actually tracking your points. We're looking at the courses that you have completed, and that helps us understand that you're following along. No. You can actually do this on any project that you want. The first week was a practice. You got assigned with a particular disease, but you also had the flexibility to choose something that you were more interested with on your own. This week, you've successfully submitted that assignment, hopefully. And if you have, now you can choose whatever you're interested in. We do hope that it will be related to some of the topics that we are doing here, but even if not, you can select any topic that you would like from any domain related to bioinformatics. And once your analysis is complete, that will be a submission that you can post, and we will do a review together. So once you have an idea, let's say you have an idea, if you were able to fill out that form, meaning you have a problem statement, you have raw data, you have done literature review that you already know how to do, that means you're ready to work on that project. We will discuss it in terms of how long will it take to actually do such an analysis, whether it's feasible and you understand that the anticipated results that you have put in there, you are going to, that aligns with the type of data that you suggested to use. Okay, so Muiwa, does that answer your question? Okay, great. And Jeffrey, so after you submit this form, that's just the beginning, right? The form is just submitting a project. And so you will get your first evaluation. After you submit that form, we'll do that evaluation in our next session, just like we did with the assignment. After you submit it and everything looks good, you start working on your presentation. Again, this presentation will be a visual presentation. Will be slides, and you kind of organize all of your slides to include introduction, to include a methods section. What is your analysis looking like? How do you actually analyze this data? Then you will talk about your results, and then you'll start working on your conclusions. After that whole presentation is done, a way to summarize your presentation, let's say you have 10 slides from your presentation, right? Then you have to summarize them in a single poster. Now that is optional. If you want to go for a poster, you want to share your poster with the world, you can do that. If you don't, and you want to just get feedback from our virtual panel of judges, you can do that. But for them to be able to address your project, they have to have something to look at. So that is a video, right? That video, when you go through your slides, explain each slide, and they can evaluate both the content, the project, and your presentation. Did that answer your question, Jeffrey? All right, excellent. So again, for those of you that are still starting to go into those projects, take a look at some of these outstanding profiles. Kylie Eckert, Michelle Namgong, and Olumuyiwa, sorry, right? So those profiles stand out. Why? As you can see, you get points for completing the coursework, and you will also get points for running those pipelines. All right. So let's talk about today's session. So, okay, when is the form due? The form is due, so think about this. We have a schedule, today's Tuesday. You have to submit your final project for review by Sunday. So you only have a few days to actually do all of your analysis. The analysis typically takes a long time. Now, the analysis does not have to be fully 100% complete for you to be able to present your data. The goal for us in this program is to help you get there as fast as possible. And I understand that, you know, some of you have other commitments and not just this program. So we totally understand that. And we want to be as supportive as possible. That's why we will provide you with guides that you can access on your own time frame and not just during the session. That's why the forum is there for you to ask any questions. And we'll also use the platform that will actually free up the resources on your computer so you don't have to be stuck doing just one thing. You can start the pipeline and continue doing other things to return back and get your results. Okay, so the form is a stepping stone. It's to help you. There's not a due date. If you don't submit it, you know, you're not graded for it. It's just to help you to get to the project. But if you want to get to the project and be able to present it, you probably have to submit this form in the next couple of days, maybe today or tomorrow. That will give you enough time to actually do your analysis. Okay, Jeffrey? All right. Okay, so let's talk about genomics in virology. And I think that genomics in virology is one of those areas where it has made a tremendous contribution. But when we think about the history of genomic data, we don't necessarily think about viruses being the primary organisms to be sequenced. So let's again review the history of genomic data. So as you know, in the 1950s, DNA double helix structure was discovered. And it was by the 1990s that the Human Genome Project captivated everybody's attention by the data that we finally discovered about the way the human DNA is organized. In 2006, Illumina became one of the powerhouses of next generation sequencing, a sequencing that used short segments of the DNA that contained the genomic information, and they had to be computationally aligned to the reference genome for us to understand the overall sequence and the changes that are observed in an individual's DNA. As a result, the amount of genomic data has tremendously grown over the past 20 years. In fact, by 2015, the 1,000 Genomes Project was finished, and by 2018, the Broad Institute announced that they completed over 100,000 whole genomes. Genomics England reported that they have sequenced over 50,000 genomes, and that amount of data continues to grow. So what type of information does this data actually contain? This information contains omics information, right? The omics information is organized by different types of data that sequencing data plays a major part of. For example, we can talk about phenomics, obviously not sequencing data, but that data tells us a lot about cell shape, how different organisms look, and what are the external phenotypes that we can observe. Genomic data contains information about the DNA sequence and the variation in that sequence. A big part of the next generation sequencing application is also around epigenetic data. That epigenetic data talks about chromatin, talks about different elements that are not in the genomic code, but that affect gene expression. And then transcriptomic data. Transcriptomic data actually talks about the expression of these genes. Now, when we typically think of omics data, we are thinking about the host. We're thinking about humans, we think about monkeys or mice. What we don't so often think about is how many viruses and microbes we interact with and live with on a daily basis. And so when we talk about viruses, actually the virome is a term, right? So remember omics, like we have the transcriptome, we have the genome, and the virome is the collection of all of the viruses that are within or around us. And the virome is one part of what's called the microbiome, and the microbiome is even a bigger part of what we are surrounded by, which constitutes all of the organisms that inhabit us, including bacterial and archaeal organisms, as well as the microbiome and other members of the meiofauna, such as protists and metazoans. Studies of the virome are in their infancy because it has only recently become possible to see the virome in large sequence data sets using bioinformatics tools that can detect relationships between viruses despite the extreme nucleotide sequence diversity. The size of the mammalian virome is not known, actually. Although our own cells are outnumbered about tenfold by our bacterial microbiome, the number of viruses may be tenfold higher still, okay? So, which means 100-fold compared to our own cells. Human feces alone contain about 110 viruses per gram. Additionally, the mammalian virome is continuously updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals. Okay, so what's important about the virome, to understand the virome, is that it is tremendously diverse. It is not studied very well, and we have only a portion of the information because oftentimes, what do you think is the problem, right? So remember, next generation sequencing allows us to sequence short reads, and those short reads are aligned to the reference genome. What do you think is one of the major challenges for us to identify and categorize all of the viruses in the virome? So again, what do you think is one of the major challenges to really know what is in the virome? Because it's hard to distinguish between new viruses and the old ones because they mutate so frequently. Okay, mutation, distinguish it from normal cells. What other ideas? Let's see. Phylogenetic relations as they mutate even between strains. Okay, that's true. The number of mutations and distinguishing them between themselves is probably a challenge. Okay, endogenous viruses have deletions in them that make them unable to express their genes. Okay, no reference genome. Okay, thank you, Emeka. That is the answer that I was looking for, because having no reference genome, we have short reads, and those short reads could be mapped only to the genomes that we know. If there are a lot of genomes that we don't know, we have to assemble them. But if we are sequencing many viruses at a time, how do we actually identify which ones go together, right? So a huge challenge is the ability to know what is the reference genome. And that is in fact also complicated by the fact that many of those genomes contain many different mutations, and those mutations make it hard for us to establish a consistent reference genome, right? So as a result, you can see how many viruses have actually been studied. So the scale of sequencing information in total here, you can see on A is the percentage growth of sequences deposited on GenBank between August 2009 and August 2010. Sequences are grouped based on the GenBank classification of division, either by their higher taxonomy or the sequencing method employed. Okay, so here you can see all GenBank sequences, right? This is percentage of increase. So you can see that this is in one sample year from 0 to about 20%. So 20% increase for all GenBank sequences in a year. If we think of viruses, you can see them right here. Viruses and bacteria were some of the higher increases, but this is what types of viruses were actually studied most. So the number of sequences on GenBank overall, HIV is the most studied, hepatitis C, B, influenza A, simian immunodeficiency virus, West Nile virus, etc. So you can see that the 20 most frequently sequenced viruses appearing on GenBank are probably the ones that cause the most severe human disease. If you take a look, we did a study in April on how many sequences were submitted for the coronavirus COVID-19. So in our study, we identified that the different outbreaks, recent outbreaks in these viral infectious diseases have been sequenced in tremendous amounts, starting from the Ebola outbreak in Africa. So in fact, next generation sequencing has been applied to a variety of different viral studies, but the most use of these has been associated with pandemic outbreaks. And Ebola virus, as you can see, from 2014 to 2016, on average per month, about 6 new genomic sequences were submitted. If you look at COVID-19, this is data from April 10th, it has over 24 genomic sequences per month. So really a tremendous growth in terms of the amount of data, but driven by actual outbreaks. So what's the difference between an outbreak and just the virome? Not all viruses are pathogenic to humans. And we have a huge bias to study things that affect us, that directly interfere with our lives, our health. So when we talk about these viruses and they just exist and affect bacteria, for example, bacteriophages is the majority of the viruses, or so it is thought, we don't actually have enough information about what they do and how do they interact with other organisms that we have studied. You can see we talked about that also yesterday when we talked about the submissions on PubMed for all coronaviruses. You can see that suddenly there's a spike in coronavirus articles with data, 158 in 2020 alone. You can see that is almost double from ever before. So this is an interesting pattern, and one of the key technologies to have more data about these viruses is next generation sequencing. So in fact, this is called viral metagenomics. So we are talking about sampling directly from environment or directly from a human. So viruses which actually most of which infect microorganisms, as you can see right here, are the most abundant biological entities, and only a small fraction of them, as you can see right here, are actually causing pandemics or epidemics or infectious diseases. And so we oftentimes think of viruses because of the viruses that cause disease. In reality, they actually grow in many different organisms, not just the humans. And even when they are in the human body, they do not necessarily cause disease. So let's talk a little bit about this next generation sequencing technology. So as I mentioned, one of the reasons for us to have an analysis pipeline that we will discuss today for this type of sequencing data is because when the data is collected in a sample and when it is sequenced, it actually has to be tied to a reference genome. And not always do we have that reference genome, right? Sometimes we don't know what are we actually trying to sequence. And that's why there are a few different techniques that we'll discuss today that allow us to study viruses and bacteria at different levels. For example, we have targeted sequencing. What is targeted sequencing? Targeted sequencing is when we are aware of what specific sequence we would like to amplify and study that sequence. Okay? For example, if I have a patient that has symptoms resembling the coronavirus, I can target and amplify that specific sequence and then do a next generation sequencing to study variation in that particular viral genome. Sometimes I don't know of a specific virus, a specific sequence that I'm looking for. And so instead, I can look for some kind of a region in all non-human genomes. For example, the bacteria and humans have different types of ribosomal genes. One in bacteria is called 16S. In the humans, it is the 18S. And so just by differentiating between those two, I can amplify just that variable region. The same goes for viruses. Viruses contain specific sequences in their genomes that are typically not found in humans. So we can amplify those sequences. A lot of times that does not provide us with complete information about what is in our sample, but it provides us a taxonomical classification, an ability to look at the taxonomy of the different organisms present in our sample. And then sometimes we want to sequence total DNA or total RNA. If you think about the human sample, let's say we take a sample from blood and we suspect that there is a virus in the blood as well. The virus is infecting the blood cell, and what does it do inside? It replicates. How does it replicate? It produces genes. So those genes are being expressed inside the host cell. If we do total DNA or total RNA, we don't deplete anything from what is being in the cell, in the sample itself. We are going to get all kinds of genomic sequences, and some of them might be from viruses or bacteria. So those are the three types of metagenomic and viral sequencing. So a lot of times, how is this used? For example, we can sequence a sample from the gut. We can know that the gut contains some kind of pathogen in the mucus, or potentially the immune cells are attached to those viral sequences. And those could be sequenced without any culturing, and then we can use it for taxonomic classification. If we do amplify, so one of the problems in next generation sequencing is that you just don't have enough DNA or RNA material. So if you do want to have enough material, you have to first culture that material, and then you can sequence based on that culture. That culture is very different typically from what you find inside the host cell. And it's very different because of the diversity. these are different organisms that interact with each other and live as communities. If you disrupt that community, you extract it from its normal environment, the composition is going to change. But what you will be able to study, instead of all of the present viral and bacterial genes or genomes, you will be able to identify specific genomes. And as a result of the rapid pace of mutation, the difference in environment can cause additional mutations to occur, and you might come to some biased conclusions because of the process of culturing. In an outbreak, when there's a viral outbreak, typically you take a clinical specimen, which is directly from the host, from the patient, and that typically includes a variety of different organisms. And you have two different approaches. One approach is to prepare primers that selectively amplify a virus sequence, right? Again, if we're looking for the coronavirus, we can create a primer specifically for the coronavirus. Or if we're looking for a flu, we can again do the same thing. We can then run and analyze individual sequences and see how they are related to the viral genome. And oftentimes, in that case, we use Sanger sequencing. Sanger sequencing is more precise, but it needs to have annotation for us to know where that sequence came from. So it is really designed around these primers. When we are doing shotgun sequencing or metagenomic next generation sequencing, we are typically taking all of the DNA or RNA in a sample. We can align some of those reads to the reference human genome or the host genome, remove those completely from consideration, and the other ones could reveal something about the sequences by mapping them on a database or by mapping them on a select few genomes that we have selected for this study. As a result, we have a lot of short sequences, right? We talked about this again. So this, for example, might have come as a result of taking a sample from a bat or a pangolin or a human, taking all of the sequences that we have and mapping them onto the host genome, removing them, and then the rest of the reads should align to some kind of a genome. You can produce a consensus sequence, and only then you have the actual nucleotide sequence from that viral genome. Now, if you do have the genome annotated, so the annotation of the genome means we know the position of individual genes, and we probably know what those genes do, and we'll talk more about that in the future. How do we know what they do? But once we know what they do, we can start looking at specific regions or sequences and identify changes. Okay, what are those potential changes? Here we have the reference genome, and that reference genome is used as a way to align the short reads to that reference. In some cases, like this position, we find a type of a change. Okay, that change is one letter, one nucleotide letter, changing to another nucleotide letter, right? So here you have a T, and here you have an A. And this is called a substitution. How many reads do we know have the same substitution? Okay, exactly. Parimala, Steparno, yep. We have 5 different reads that are all in agreement that this is an A and not a T. Okay, in another example right here, what do we have? We have a G and an A, and in the reference we have a C. So what do you think? Can we accurately determine what is the mutation in this position? Right? It's going to be difficult, right? So we actually cannot decide whether it's a to or a C to an A or a C to a G, right? We cannot decide that. And the reason for having more coverage, which is what this is called, is coverage, is because of these kinds of situations, especially if we have a lot of mutations occurring all the time, and those mutations are of different kinds. So here you have two examples of substitution. Here's an insertion or a deletion. How do we know this is a deletion? Here we have blinks as opposed to this sequence in this region of the reference genome. So to avoid these kinds of ambiguous changes, we need high coverage, right? And high coverage is just the number of reads in that specific position. Okay, so to do next generation sequencing, there are three common formats that are typically used. Those are the FASTQ, which are the individual reads, and those reads are short. They can be anywhere between 30 and 300 base pairs long. The FASTA is typically used as a reference genome, so we actually map the short reads onto the reference, which is included in a FASTA format. And then we also have the annotation. The annotation is oftentimes included in what's called the genome transfer format, or GTF, and also sometimes called GFF. Okay, so that includes the information about each particular gene or exon or any kind of a genomic element in our data. Here's an example of a FASTA file. You already know what a FASTA file looks like. Okay, so the FASTA files contain continuous sequences that are not broken up. If they are from a big, large organism like a human, they will be broken up into chromosomes. If the data is not assembled in a single genome, those will be called contigs. Okay, they're assembled in bigger chunks, but not the full genome, and those are called contigs. The FASTQ file, who can tell me how long is a read in this FASTQ file? Yep, Parimala. Exactly, 160, right? So we can see right here, the length is 160. So there are 160 nucleotides. And those nucleotides are one single read. And there's many, many reads. You can see that each one has an ID. So what happens to these reads? These reads are aligned. Okay, so here you can see reads are aligned. And once they are aligned, this alignment is called a BAM file. Again, there are two formats, BAM and SAM. So the BAM file and the SAM file contain the exact same information. That information is exactly the position of the read, where it is aligned, and how does it align to the reference genome. Kind of like multiple sequence alignment, but instead of aligning multiple sequences to each other, it is just aligning of the reads to the reference genome, okay? So how can we know what are those alignments? Those alignments could be visualized. For example, we can use JBrowse or Integrated Genome Viewer or UGene. Some of these programs just provide us with an ability to see what is for each individual read and the alignment of the genome in terms of the BAM and the SAM files. Okay, so what do they look like when we visualize them? They look like this. Okay, so do all of you have access to the server platform, right? Let's take a look at this example. Please go with me to server.t-bio.info and we'll look at an example together. Okay, let me know when you are here. Okay, just type in yes when you're here. Okay, I'll let everybody get there. Okay, anyone else? All right, great. So now we can go to either two sections. I'm pretty sure you probably should already have been seeing this, but you have here the option to open up each section and look at an example, a demo pipeline from each one of these, okay? So we can go to mutation variant, and we will see there an example, and we can also go to this RNA-seq project, and we'll see an example there. So let's take a look over here, right? Demo variant calling cell lines. We'll click on start first, where we'll see the data, the link. The link is right here. So tell us, Parimala, when you're here. Okay, Parimala, tell us when you're here. So we'll start over. Did you get there, Parimala? Do you have the link? All right, great. So once you're here, go to mutation variant. You can always hover over this eye to see what each section is about. So you go to mutation variant, parallel analysis of mutation variant data. Okay, you go to variant calling cell lines. You click here. Then we click on start. Okay. The alignment. So we align the reads to the reference genome. And then we can click on visualization. Visualization uses JBrowse to actually show us individual reads. How do they align to the reference genome? And that's it. We just click on end. And then you should see this visualization link. And you can open that. Okay, so once you open it, it will take some time for it to load, but you should see something like this. Okay, if you have it, please let me know that you got there. Okay, so if you see something like what I see, you can just say yes, and that you got there. Did anyone get here? Okay. Okay, so let's do this quickly once again. So it's not the same password as on the main site. It's a different password. You should have gotten that password by email. So Suparno, if you can look in your emails, you should have gotten an email from marketing at Pine Bio from Bipsa. Okay. So Bipsa, I don't know if you could maybe email him. Okay. Okay. So Karina, you go to NGS data, right? So we're doing NGS data. Then you go to mutation variant, parallel analysis of mutation variant data, and you click on this down arrow, and you click here. Okay, Karina, did you get here? All right. So then we click on Start. In Start, you can actually see what are the input and output files. Well, in this case, you don't see it, but typically you would. Go to Bowtie 2. Okay, here you can start seeing those. So Bowtie 2 loads the data and aligns that data to the reference genome. After it aligns it to the reference genome, we just click on Visualization, and you click on End. So Parimala, it's a different ID. It's not the same password. Please look in your emails. You should have gotten an email from Beepsa on using this pipeline. The title of the email is tbioinfo is the subject line. Okay, so Parimala and Suparno, please find those and we'll try to email you. Okay? So here, you just click on Start, then Bowtie 2, then Visualization. And then you click on End. Okay, Suparno? Once you do that, you will see this visualization link. Okay, the visualization link, you click on it, well, it takes some time for it to load because what do you think is this reference genome? Who can tell me? What is the reference genome that we're using here? Okay, does anyone know what is this reference genome? How would you know what is this reference genome? Okay, any ideas on what might be this reference genome? Okay, Chandrani, thank you. What is the number of chromosomes that we have here? Well, yeah, this is chromosome 1, but how many do we have in total? Okay, you click on here. How many in total do we have? 23, okay, so who has 23 chromosomes? Okay, so the reference genome here is human, right? Okay, so we have the human reference genome. And if you zoom in, I wanted to zoom in and just show you that you can actually see the sequence, right? So continue zooming in. Here's the sequence, right? We have the two direction of strands, and then we have the frames that convert it to amino acids. And then we have the actual data. So our data is right here. These are the reads. And this is the reference genome, right? So now let's zoom out. Okay, let's just zoom out until you see these individual signs right here. Okay? So what are these little signs? Let's zoom in. Right, well, don't go out too much. Just go a little bit. And what are these individual signs? What is chromosome M? Mitochondria. Okay, so if you go through and you zoom in, again, don't go, if you go out too much, you will have too much data. Remember that this is actually loading all of the available reads, and look how many nucleotides we have here. In chromosome 17, we have over 80 million nucleotides, right? So it will take a long time for it to load. Okay, so remember that. And this is using RAM from your computer. Okay, so if you don't have a lot of RAM, it is not going to show you. Okay, so play around with this. You'll see some more information on this in Genomics 1. Let's move on. Let's see what are actually some of these positions that we have found. Okay, so this is just an example from that very same example that we are looking at. Again, as I mentioned, if you zoom to a particular region where you can see this is the reference genome, right? This is the change, so the changes in the reads that we have from the reference genome. Here you see G, and here you see A, right? So the reference is G, or sorry, the reads contain a G, and it's in 100% of the reads. Okay, you see here 3- and 3+. What do you think that means? What do you think is the 3- and the 3+? Any ideas? So what are those plus minus? So what is the total number that is different? It's 6, right? But why don't we have 3- and 3+? Okay, the answer is, as you can count the reads, 3 are blue, right? And 3 are red. What do you think are the red and the blue? The red and the blue, right, they're positive, negative, because we have what's called pair-end reads. Okay, what are pair-end reads? Pair-end reads are reads that are sequenced from both ends at the same time. So they're single-end reads and paired-end reads. Single-end are just one direction. Paired-end, they take each segment when it's shattered and they sequence from both sides. Okay, so that's why we have blue and red. Now here you see all of those contain a G. It's 6 in total compared to the reference A. So it changes the amino acid. You can see the amino acid is M. Okay, so 3G is not there, or in 3G is present. Well, no, in all of them, in all 6, the G is present compared to the reference. Okay. So Parimala, I think Bipsha is looking into this. Bipsha, did you figure out what the issue is with the password? You can let Parimala know. For the rest of you, right, so what are these called? These are called SNPs, single nucleotide polymorphisms, okay? They change, and here we have a change that we've identified, and some of them have a stronger evidence than others. For example, these individual ones, okay, so for example, there's a G right here, there's a T right here, right? But it's only in one read, so it is not significant. So you don't see a sign like this there. Okay, so does all of this make sense? Does anyone have any questions about what we're seeing in this visualization? Okay, so if there are no questions, what types of mutations are there? There are at least 3 different types of mutations. Missense mutation. What is a missense mutation? Missense mutation is when a single DNA base results in the in a change in the amino acid. So if we go back over here, these ones, right? So these ones that are highlighted over here are going to change the amino acid sequence. Sometimes they don't change the amino acid sequence, right? So like here we have a codon change and that changes the amino acid sequence. Sometimes they do not. Nonsense mutations actually create a stop codon. Who knows what is a stop codon? Well, you don't need to tell me because it says it right here, right? So it terminates translation. So in other words, that protein can be shorter and not function. And then sometimes we have a frame shift mutation. So instead of having one sequence, it will have a different sequence, but all downstream. So the downstream sequence will be different. Those are the different types of mutations. And we can observe them by only looking at those places where we have significant evidence of the changes from the reference genome. So this critical step of alignment of reads to the reference genome allows us to discover different types of mutations. Okay, are there any questions about the process of sequence alignment and the process of identification of mutations when we do have a reference genome? Again, remember, in that case, we looked at one particular genome that was the human genome. If we know what virus we want to sequence and we have that genome, we can align the reads to the virus genome also. Okay, are there any questions? Okay, Perimala, did you get access? Were you able to use the password that Bipsa emailed you? Yes, you would have to install it. Okay, so if you search for JBrowse, you can definitely find it. So JBrowse, right here, you can download the latest release. This is the link. There are also other places where you can access, where you can read the SAM and the BAM files. So you can go here, for example, if you look, this is the SAM file from this particular alignment. So you can actually download this. So there are two samples here, two SAM files. If you download them, you can use JBrowse, you can use Integrated Genome Viewer, you can use another software called UGene that allow you to view these kinds of FASTQ files and SAM and BAM files. Okay, so please take a look at these. Parimala, were you able to log in? Have you tried checking that? Okay, so you got the password. Well, okay, so you needed to have a SAM file, correct? And then you don't upload them on JBrowse. JBrowse does not allow you to upload anything here. You have to install it. You have to have some place that runs it. Okay, so what we have, so you can see JBrowse Desktop, for example, runs as standalone app on Windows, Mac OS, and Linux. Okay, and that's all this does, is it actually takes this file and visualizes it in JBrowse. So if you process your data, typically you don't start with a SAM or a BAM file, you start with a FASTQ file. And FASTQ file is generated from Bowtie. You can also install Bowtie on your computer and run that. And once you generate the SAM file or a BAM file from Bowtie, you can actually visualize it in JBrowse, Genome Viewer, or UGene, for example. Okay? So Parimala, do you want to just try once again, or do you want to share your screen quickly and we can all take a look and see how we can fix this issue? Okay, well, I'll let you know, if you want to connect later, we can try doing that together. Okay, so that information, right? Well, okay, so Bibsa, maybe you can speak to Parimal about this. Okay, so if there are no other questions, what do we do if we don't know what is the actual genome that we are sequencing to? In another section, in metagenomics microbiome, we have an option to use, again, bow tie, but instead of using the individual genome as a reference, we can use a whole database. So which database can we use? On NCBI, there's a specific database for all of the viral genomes. So we can actually use this information and map our reads on all of those genomes, right? There's thousands and thousands of genomes there. It's called RefSeq, reference sequence. And you also have an NCBI virus visual interface where you can explore what kinds of viruses do they actually have. So if you go over here to all of the viruses, you can see that there's about 219 different sequences. All of these are full genome sequences, or they could be partial sequences. You can see partial here, and they contain some annotation. Where did they come from? How they were generated, etc. So we can map on all of this sequence. So if we go to the metagenomics section, we can actually input the raw sequence of those reads, align them to the virus genomes. And when we, okay, so Parimala now is back in. So Parimala, just quickly, sorry for everyone else, we'll just quickly go through this so that Parimala can follow along. So Parimala, logging you out. So are you in now, Parimala? Okay, so can you go into this main areas of analysis and go to mutation variant, parallel analysis of mutation variant data? Click down here on this drop down, okay, and click on this demo. Tell me when you've got in. Now you click on start. After start, click on bow tie 2. Okay, and then click on visualization. So you click on visualization, you click on end. And you should see here a link to visualization. Let me know if you got there. All right, great. When you click on visualization, you'll be able to see that visualization that we were looking at, and you'll be able to explore by zooming in, zooming out, changing the chromosome, and focusing on a specific portion of that genome. Okay, so another option, let's do this together. Let's go to areas of analysis. And now instead of mutation variant, we will look at metagenomics microbiome. So you click down here, and you have demo metagenomics regular. So please click on this demo. Okay, let me know once you're here. All right, so let's click inside. Everybody else, please tell me once you are here. Okay, I have Kylie here. Anybody else is here? All right. All right, so remember, this section deals with a whole database of genomic sequences, not just one, but many. But it's the same idea. So we actually are going to take some metagenomic sequencing, and we will map them onto a reference database. So here, bow tie to align can use different types of sequences, different types of databases, sorry, and align to all of those, and then calculate abundance, right? So remember how we looked at reads? Some reads will align to different genomes with different abundance overall. Okay, once we do that, we can also replace those names from having just a general name, having an annotation, then click on end. Okay, and we get some output. So what is going to be the output? The output is going to be the result of this annotation. Okay. So again, we're not going to look into this too much today. We're going to look at this tomorrow, but just wanted to show you how it's done. Let me just quickly show you another example, this time in the transcriptomics section. Okay. So for those of you that are still here, tell me if you can go back out. Okay, and we'll go to, again, this first one, RNA-seq cell line project. Okay, and again, how to go to the output. You download the output to your computer. Okay, so I'll show you as an example. Here I click on start. This is for genomic data analysis. But here I will just visualize data. So here I'll go to bowtie 2G, visualization. Again, this is going to be using this specific JBrowse, right? And I have visualization. I also can download some of these files that will provide me with some information. I can just open them on my computer. Okay, so if I go to visualization, again, what do I get? I get a different data set. This is not the human genome. This is a different genome called SF9 cell line. Okay. So here you will see that your reads are loading. Let's zoom in. Okay, here we have more of those reads. Right now we're just looking at demo pipelines. So if you select, you can select both a regular pipeline or a demo pipeline. If you select a regular pipeline, you will be able to do analysis. But right now we're not doing analysis. Right now I'm just showing you a few examples. Okay, as you go through the courses, there will be assignments that will explain to you what to upload and how to actually process that data. Does that make sense, Aparno? And again, we're not an RNA-seq chip main pipeline, right? So if you go to your areas of analysis, you have to open this drop down, right? So instead of clicking here, you click right here. Okay, so what do we see here? Let's zoom in. Okay, you can see that many of these are actually in areas with little coverage. But here we have something with significant coverage. Let's take a look at what we have. Okay, so what is this? Let's take a look. Okay, what do you think this pink, or sorry, the purple line is? Let's zoom in. Okay, who can tell me what this is? Yes, but what kind of a mutation is this? Okay, take a look at where my my mouse is. What does this say? Okay, great. Yes, so Chandrani, right? It says the majority of the reads, 82% of the reads are actually insertion. And there are two other substitutions. There's a substitution in 7 reads, or 8% substitution, and another substitution in 1%. Okay, so some of the reads show a substitution, some of the reads show an insertion. Okay, so you can see here different types of mutations. Some mutations agree in evidence between all of the reads, and some of them don't agree on all the reads at all. What you can do is you can also click on an individual read and actually find that read sequence and quality for each one of your nucleotides, okay? So again, the problem here is to do this kind of analysis is that you have millions and millions of reads for each one of your samples. And here we're just looking at one sample at a time. This one sample is an alignment to a particular genome, right? So when we do visualization, we can only do visualization on one particular genome. Before we decide which genome to align our data to, we have to, what we did in our other example, actually align it to a database sometimes, finding what other genomes are there, right? And these could be bacterial. So you can see here bacterial genomes, 16S, which is, we'll talk about this next time, or viral genomes. Okay, what is an example of the output? If you actually download the output will show you the annotation file. In this annotation file, you will get the accession number from NCBI for different types of viral sequences and how many reads align to that genome. For example, here in this one, we found some reads aligned to the Ebola or influenza or some other genomes. You open it from, so again, you need to be, okay, let me do this again. So what we did was this one, right? Suparno, maybe you can follow along. Okay, so I click here, I click on start, bowtie 2G, visualization, end. So I have to click on end, and then I should see a link to visualization. That's how you open the JBrowse page, from this link. Did you find it? All right. Did everyone else find these? So who successfully found all of these different visualizations? Okay, Karina, great. Anyone else? Megan, okay, perfect. Anyone else? Did anyone have any challenges that we can address while we're here? Okay, well, let me know if you face any issues. So here, in this example, I just gave a couple of examples of different types of samples. Okay, the first sample I showed you was a sample of a human sample from a human cell line. The second example was an example from an SF9, which is a Spotiptera frugiperida, which is a different organism. In fact, what I wanted to tell you next is that there's a project called Spotiptera frugiperida, SF9 cell lines. It is a cell line that is used for biologics, production of biologics. Who knows what biologics are? Okay, enzymes or proteins, yes. So these are, right, so they're drugs based on, exactly, drugs based on some kind of a protein. So whenever you want to produce a vaccine, you want to produce some kind of an enzyme like insulin, for example, you can use a cell line that produces them. So you can actually engineer a bacteria or a virus that will produce a specific thing that you need. But of course, you need to make sure that that cell line itself is not contaminated with viruses. And so a big part of what the FDA does is it checks all kinds of commercially available cell lines to make sure that those are not contaminated. And this is a project based on a publication where the FDA was actually checking through some of these cell lines, and these SF9 cell lines were detected to have some contamination. So they were studying what type of contamination they had, and they found a contamination with a particular virus. And that was a new virus that previously was actually not identified. So let me show you where to find this project. Okay, if you go right here to projects, right, for those of you that would like to understand this a little bit better and see the application of this type of analysis to a real world situation, you will find this project right here. So this project will guide you through that analysis, show you some of those example inputs and outputs, where to find them, how to process them, and what would be the outcome. Okay, also a good idea for someone who does not have a project idea. You can kind of see what that project idea might look like. Okay, another example that you will find in Metagenomics 1 is how to apply this mapping on a database to identify taxonomy between different types of samples using metagenomic sequencing. Okay, and this is going to be relevant for our session tomorrow. So your assignment for tomorrow is 3 things. Take a look at the project, the SF9 project. Take a look at this blog article that we have on different resources for viral genomic data sets related to COVID-19. So that blog will tell you about some additional resources where you can find data, explore data. Some of you mentioned you want to do an analysis of COVID-19 data, so that some of those resources are listed out there. And Introduction to Metagenomics. That course, sorry. One second. Okay, sorry about that. That course is designed to introduce you to what we'll study next time. Okay. Okay, so again, we talked about viral outbreak, the course in between. This is not a course, this is a blog post. Beepsha, can you please paste the link to the blog post? Okay, so we talked about sequencing of these viral outbreaks. Viral outbreak sequencing typically relies on filtering out non-viral reads. So that example is provided in the project, the SF9 project, using that particular cell line. We can apply the same technique to study, for example, presence of viruses. Okay, Kylie is asking, do you have to do the intro to metagenomics? Again, you don't have to. If you want to learn a little bit more about metagenomics and different types of metagenomic sequencing, you can, but of course you don't have to. Okay, and then we can apply the same approach to study. Yeah, that's fine. You can do the same analysis in application to COVID-19. For example, if you have a human sample that is infected with COVID, you can take those cell lines, map your whole transcriptome onto the human, the host cell line, and then you will get non-mapped reads, right? So those unmapped reads, the process of getting those unmapped reads is explained in detail in this SF9 project. And then map those onto the reference genome, right? If we are suspecting that it is infected with this novel coronavirus, you know how to get that FASTA file, right? That FASTA file is on NCBI. So you can actually get that FASTA file, map the reads, the unmapped reads onto this coronavirus, okay? So tomorrow I'll actually walk you through this pipeline so that you can take a look at what happens when we have contamination with a virus, but we only have gene expression data from our patients. Now, what's important is that these viruses, as I mentioned yesterday, cause a whole variety of different diseases, including cancer. And oftentimes, when we have this sample from the host, we might be interested in looking at what is actually going on, and we can map the reads onto these different viral genomes to really understand what's going on. So let's take a look at an example. Here we have a data set. Okay, what kind of a database is this data set from? What kind of data is this? Okay, this is on NCBI. So what kind of data set is this? Okay, Chandrani, thank you. It's a geo data set. So what kind of data is this? Is this genomic data? Is this metagenomic data? Is this transcriptomic data? What kind of data is this? Okay, thank you, Chandrani. Transcriptomic data, right? So everybody is following along. We have here geo, right? It's listed right here on gene expression omnibus. So this is transcriptomic data. What did they do here? They were trying, what is a geo data set? So we talked about this yesterday. Geo stands for gene expression omnibus. So this contains expression of genes. Okay, Paramala, does that answer your question? Okay, so here you can see what were they trying to do. Next generation sequencing identification of HPV, which is hepatitis B virus, MLL4 integration in its molecular basis in Chinese hepatocellular carcinoma. So this is a liver cancer. And they were trying to look at, sorry, the virus inside the human cells, right? Here's what that data looks like. And we can take this data. So this is the SRRs are the individual samples. So you can see those samples are taken from tumor, non-neoplastic liver tissue, right? So this is like a tumor and a control from the same patient. So as a result, we can take a few examples. So here's 3 examples. One is a tumor, another one is a tumor, and one is a non-tumor. Let's see whether we can identify here a presence of this hepatitis B virus. So what would we do? We would go to RNA-seq section. We can build this pipeline. What would you use as a reference genome if you're trying to identify whether that virus was present in those samples? Right, HBV, right? So this is the hepatitis B virus, right? So HBV. So we map those reads onto the hepatitis B genome. What do we get? So here we have our 3 samples, or this is just 2 samples. So this is 637 and 642. Okay, let's take a look. 637 and 642 at the end. Okay, so 637 and 642. So this is a tumor and non-tumor. So we are comparing them. And what do we see? Okay, so here we have 642 and 637. What is the difference between them? Who can tell me what is the difference between these two samples and how they align to the reference virus genome for hepatitis B? (642) has more closeness to reference genome. Okay, based on what? How did you determine that? Okay. Well, let's take a look at more reads for the one on top, says Emeka. 637 is a tumor genome, 642 is a non-neoplastic one, yep, the non-tumor. But let's take a look. So here we have our scale, okay? The scale is different. Here it is 20,000, here this is 2,000, okay? So the difference is tenfold from here. So that's one difference, right? So one difference is that this is at about 10,000, this is about 1,500, okay? The other difference, as you can see, is that this stops right here, okay? So who can tell me where do I get the information? The scale is for coverage. So how many reads? Okay, remember we looked at those reads. They look at the individual reads, but that's a lot of information to show. You see it says too much data to show. You can't look at the individual short reads. So instead you're looking at all of the reads in terms of coverage. Okay, so the coverage for the top one, how many reads are mapped onto the top non-tumor sample, is less than how many map onto the bottom one. But it is not consistent throughout the whole genome, right? This is the whole genome. As you can see, this whole thing is covered. How do we know which regions have more or less coverage? What file gives us that information? What file do we use for annotating the genome? Okay, we use GTF. All right, excellent. Emeka, so in this case, this is not a GTF file. This is the GFF file, but it's the same thing. And you see it's the GFF3 Hep B virus genome. So that is the information right here, GFF3, right? So the genome annotation is in a different file. That annotation includes the beginning and the end of each one of the genes, okay? So whatever we know about this genome are the genes. And here you can see all the different genes. So if you look here, right, you can actually draw a line and see that this portion is an additional gene, right, signal peptide, that is not present in your non-tumor sample. Okay, so there are two differences. One difference is coverage. Another difference is which genes are actually being expressed in each one of your samples. Okay, one gene from the whole viral genome, the viral genome does not have that many genes, has only a few. For example, the coronavirus has 29 different genes. This genome also has only a certain small number of genes. So each gene plays a very important role in whatever the virus can or cannot do, including interfering with the host immune response, including the ability to produce the final virion to be infectious, to spread between different cells. So it has a huge role. Okay, so how do we know that the virus is there. The virus is there is if reads map onto a significant portion of the genome. How do we know that this is present and replicating? The more this is covered, the more the coverage, and more genes are being covered, the more we have a higher confidence that this is actually happening. Okay, in many cases, we can also simplify this because here we are not taking into account that some of these reads could actually map onto the host genome. We're just assuming that whatever comes onto and maps onto the viral genome is in fact from the viral genome. But to make sure, remember, we have to first eliminate the reads that do not map onto the host genome. And we can do that by mapping first onto the reference host genome, which is human in this case, measuring the expression, and then we can take the unmapped reads and map those onto the hepatitis B virus genome plus the annotation. Okay, here we take these two samples and let's run it again. And we can see that this difference is actually amplified. Okay, so you can see the same thing, but the end is right here. Again, the difference becomes, the scale becomes different, right? But what we see is this important signaling peptide being present in the infected tumor sample. Okay, so again, a lot of this information is contained in the data. The analysis, however, is key to be able to extract relevant information to the question that we might have. And when we look at this question that we might have, it's important for us to understand that the data will provide us with that evidence. Yeah, so, you know, I'm not just asking the question, do they match? I also want to understand how well do they match? I want to understand which genes are being expressed from a virus. I can now ask all kinds of questions based on whatever data I have. I also need to understand what exactly was sequenced, how was it sequenced, what do I expect to find in this data, okay? So that's an important part. This is just the beginning of that analysis. And hopefully by going through the SF9 project, you'll see some additional steps. For example, one challenge is just identifying presence of a virus. Another one is to determine whether that virus is the actual hepatitis B or whatever other virus you suspect it is, or maybe it is something else that is just a little bit different. For example, the coronaviruses have a similarity of 98% between bat coronaviruses and human coronaviruses. Okay, so 98% is a large proportion, but the 2% that don't match play a big role. Okay, so there are many questions when it comes to these genomes. All right, in our next session, we will talk about microbial communities. So instead of looking at virome, we will look at the metagenome or the microbiome. And the microbiome can be also identified using a different method. Instead of looking at the full genome, like we looked for the viruses today, we will actually look at the hypervariable region. We will take a look at how we can use specific sequences that are different between humans and bacteria to understand the composition of the microbiome in a given sample. Are there any questions about what we did today? Did you have any questions, or did I lose someone in this explanation today? Okay, so everything is clear for everyone? Excellent. All right. Okay. All right, so Parno, great. So hopefully tomorrow you will let me know how it goes. Maybe you will be able to try some of these pipelines that we discussed. Again, before trying to run a pipeline, take a look at some of the examples. When you look at these examples, this is an example with a description. There's a course that provides you with some, evaluation steps, or you can just go here and go to the different demo pipelines and just play around with them and discover what are the inputs and the outputs. So again, every time you see this little drop down, you can probably find the demo pipeline that you can play around with. Now, the demo pipelines don't count as points, but you will be able to see what's the outcome of a pipeline, what are some of these outputs, download them to your computer, make sure that you understand the steps, and hopefully those pop-ups explain pretty well what the steps are. Many times they use open source software, so if you want to replicate the same on your computer, you could. You would have to install that information and make sure that your pipeline works together, but it gives you a good insight into how pipelines work. What do they do to give you meaningful outputs? And then we will also talk about what to do with those outputs. We'll actually discuss how to evaluate those outcome files. Thank you for joining today. Again, if you have any questions, please put them on the forum. We will keep updating you there with any kinds of feedback. And if you have your video presentation from your assignment from last week, there's still time to submit it today. We will be emailing Sona everything that was submitted for a review. And if you have any additional changes after that and you want to use that information for your personal project, you can definitely do that. If you don't, then please start submitting your projects on the forum. All right, thank you everyone, and hope to see you again tomorrow.